I am Lara Campana. I'm the Senior Vice President for Research and Translational Science at Resolution Therapeutics, and I'm one of the co-founders of the company. At Resolution Therapeutics, we develop regenerative macrophage therapy for inflammatory and fibrotic diseases. And the reason why these cells are so exciting is because they naturally appear in all our bodies exactly with an anti-inflammatory and an anti-fibrotic function. So they are naturally endowed with the key pharmacological abilities that we need in order to tackle the driving pathological mechanisms in inflammatory and fibrotic diseases. An example of which can be end-stage liver disease, where patients have a chronic liver disease that results in scarring, so fibrosis, and chronic inflammation, and where regenerative macrophages can be particularly suited to tackle these challenges. We're often asked why regenerative macrophages rather than, for example, hepatocytes, the natural cells that populate our livers and carry out most of the metabolic functions. Well, the answer lays in the microenvironment. So when a patient has a chronic inflammatory and fibrotic disease, such as end-stage liver disease, the liver microenvironment is dysfunctional. There is a lot of scarring, which almost strangles, for lack of a better term, the resident cells. And there is inflammation, which triggers a toxic environment for the resident cells. So even if we decide to put in fresh hepatocytes from a laboratory, these cells will still be subjected to the same microenvironment, which is hostile to liver regeneration. And therefore, they will be subjected to the toxic effect of the environment, most likely die, fail to engraft. And actually, this is not only a theory, but supported by data. We have been trying as a hepatology community to do liver transplantation for over 30 years now, with very poor clinical outcomes and no clear path to a therapy. So regenerative macrophages are a better option because instead of trying to replenish the resident cells with a microenvironment that does not allow them to thrive, is an approach that aims at restoring a permissive microenvironment so that the endogenous cells of the patients can now promote regeneration and restoration of a certain level of metabolic function. Regenerative macrophage therapy was first born at the University of Edinburgh in terms of academic findings. Professor Stuart Forbes conducted for many years research on the fundamental mechanisms of liver regeneration. And the wider liver group in Edinburgh identified this population of resident macrophages that naturally occurs in the liver of mice and then humans also during the regeneration phase. And it was then thought, well, if these cells do exist, why don't we try and take them to patients in order to improve liver regeneration? So this is what at the University of Edinburgh, Professor Stuart Forbes and Professor Campbell developed as the so-called matched clinical trials, in which patients with stable cirrhosis were treated with a sort of initial version of this regenerative macrophage therapy. The process was quite simple. We took monocytes out of the blood of the patients and then converted them utilizing a cocktail of cytokines to regenerative macrophages, then to be reinfused in the patient themselves. At this stage, this was a proof of concept for tolerability, which went actually really well. Our phase 1 study did not give us any concern around the tolerability of the therapy, which allowed us to progress to a phase 2 study, which is now already published in terms of one year data. And this study was the springboard on which we decided to spin out and fund Resolution. Whilst Professor Forbes and Professor Campbell stayed as advisor, what I took into Resolution in terms of leading the science and the strategy in the scientific strategy in the company was their attention to scientific rigor and really the passion for treating patients. They are two people that are very passionate about this patient population, and building on this passion for treating this patient population at Resolution, we decided to go for the patient population in the field that has the biggest unmet need, the so-called decompensated liver disease patients. Those are patients that not only have a chronic liver disease, but they already developed very severe events, clinical events that require hospitalization, such as gastrointestinal bleedings, ascites, which is accumulation of liquid in the abdomen, and then encephalitis is a problem with their cognition and brain. What I've also took there is the attention for the mechanistic aspect, and this is where Resolution decided to develop and build on their experience and transform an academic proof of concept into a medicine by treating a more severe patient population, but also by engineering these regenerative macrophages to improve their anti-inflammatory and anti-fibrotic functions. The Resolution approach to engineer macrophages is actually a very gentle one. So what we do is that we insert RNA into the cells to encode for IL-10 and metalloprotease 9, also known as MMP9. This is a very gentle approach because it doesn't entail any genetic material entering the nucleus, thereby also lowering safety concern around the engineering of the therapy. And we have an engineering method that preserve over 90% of the viability of the cells. So it's very respectful also of the cell health. And the reason for us to seek an engineering is to improve the anti-fibrotic and the anti-inflammatory functions of the cells, which are key to their mechanism of action and driving of clinical benefit. And after an in vitro screening that aims at measuring some key parameters around the anti-inflammatory and anti-fibrotic functions, IL-10 and MP9 were the two genes that came out as the best in class, and hence why we then combine them to generate a more potent regenerative macrophage therapy for use in this patient population with a high unmet need and no currently available medical therapies. Currently, Resolution is running the Emerald clinical study. This is a phase 1/2 study that measures the various parameters. First and foremost, the safety of RTX 001 in patients with a decompensated liver disease. We also have a measurement of clinical efficacy in terms of the ability of the medicine to reduce decompensation events, so these very severe events requiring hospitalization, but crucially in preventing death and transplantation. Unfortunately, in this patient's population, once the decompensating events start to appear, the median survival drops drastically to 2 years after one decompensation event and 9 months after 2 decompensation events. So the ability of the cells to prevent further decompensation event, death and transplant is a key parameter to measure their clinical potential to become an approved medicine. And on top of that, we're also collecting some blood biomarkers that will help us really understanding into the finer details how this medicine works in patients and therefore how this medicine prevents this progression of the disease. So the MATCH study is the academic study that we run as University of Edinburgh. This was a phase 2 study. It was a randomized control study, which means that we had 2 groups of patients, one receiving the cells and one receiving standard of care. It's important to note 2 things here, that these patients were a stable patient population, so a patient population that's factually less severe than the one we're targeting with Emerald. And second, that these patients receive 1 dose of cells, whilst in our engineered clinical study Emerald, we're giving multiple doses of the cells. This initial academic study was designed as a proof of concept that regenerative macrophages could be utilized in patients with advanced liver disease. So once again, safety was at the forefront of the thinking. And again, this phase 2 study showed that the non-engineered cells were very well tolerated in this patient population. Furthermore, we also looked at clinical events such as those decompensating events, but also death and transplant, and we could see an advantage in the treated population at 1 year. And we're now in the process of submitting and publishing the 4-year follow-up data. So watch this space for that. If someone is out there thinking, well, I have an innovation, I still work in academia, and I think that this innovation could be a real benefit to patient, whether this is a medical device or whether this is a medicine or a diagnostic tool, my advice would be do it. Get out of academia and do it, because there are two things that are universally true. If you really wanted to change the lives of patients, you need to have a product of whichever nature, diagnostic, medical device, or medicine. You have to have it approved. And to take medicine, for example, to approval, it takes a village. So it takes a plethora of skills that range from finance to regulatory to engineering to biology, and it takes capital, both of which cannot be found strictly in the academic environment. So if you're wondering whether there is a fantastic science out of academia, yes, there is, and there is also science that can have real impact on patients. And if you're passionate about having impact on patients, take your proof of concept. Much for us was a proof of concept, would have never been a medicine, would have never been a product, and take it out of academia and make sure that you surround yourself of competent people that can help you having that impact you want to have.